Hello, everyone, and welcome to today's webinar. And today's webinar is titled, just bear with me 1 second, is titled Demonstrating Analytical Comparability in Analytical Development, hosted by Biopharma Webinars and presented by Santoshkumar Khatwani, Director of Analytical Development at Sangamo Therapeutics. So, Santosh, hello there, welcome. Thank you for joining us today. It's a pleasure to have you.
Hello, everyone. Thanks for joining today. Today, I'm going to talk to you about some of the concepts of comparability. But before, I would like to thank Biopharm Asia for invitation, and also thank you all for joining.
Thank you. Also, Santosh, just going to pause you there 1 second as well, just to introduce as well James Wilkinson. So, hey there, James. Thanks for joining us as well today. It's a pleasure to have you. James is the Vice President of Corporate Development at Refeyn. So, hello there. And I was just about to introduce Santosh, so to tell the audience a little bit about what you do, if that's okay with you, Santosh, as well. Yep. So Santosh is currently serving as Director of Analytical Development at Sangamo Therapeutics, and Santosh received early education in India with bachelor's degree in chemistry, followed by a master's degree in organic chemistry. He joined National Chemistry Laboratory in Pune, India, and worked on synthesizing organic compounds for potential pesticide applications. Later, he moved to the United States to pursue a doctoral degree, PhD in chemistry from the University of Kentucky, where his research focused on designing biosensing systems and responsive biomaterials using chemically modified proteins. And furthermore, he joins the Department of Chemistry at the University of Minnesota to conduct postdoctoral research in the development of chemical and enzymatic approaches for generating novel protein DNA complexes. But Santosh, over to you. Please don't let me keep you any longer. Welcome.
Thank you, Marcus. As I mentioned earlier, I'll talk to you about some of the general concepts of analytical comparability, especially how to deal with that in analytical development group, and also some of the considerations that you may want to look into before you start these studies. But before I go into my talk, I want to give you a brief background about Sangamo Therapeutics. We are headquartered in Brisbane, California, and we have sites in France as well. And as you may see here, we have a diverse portfolio of clinical and non-clinical candidates. Some of the programs are wholly owned, are shown on the left, and there are several programs that are partnered, are shown on the right. In the wholly owned programs, we have a flagship program of gene therapy in targeting Fabry disease. It is currently in phase 1-2 trials, and we mentioned several months ago that we intend to pursue phase 3 pretty soon. We also have a program for sickle cell disease in currently phase 1 trials, which focus on cell therapy application. And we also have a phase 1-2 program in renal transplant for TX200 cell therapy program. And we also have several non-clinical candidates that are shown below that. And among partner programs, we also have a flagship program of hemophilia A with the Pfizer, which is currently in phase 3 trials. We also have several other programs with the Biogen, Kite, Novartis, and Takeda that are shown also in the slide. This is the outline of my talk, and I will just give a brief background about general considerations of analytical comparability. And I would like to ask 3 different questions: why, what, and how, with respect to analytical comparability. What is the typical process for the demonstrating such comparability? And also, what are the typical contents of the protocol that are typically written before you execute the comparability study? And since we work in gene therapy applications, and one of the candidates widely known in gene therapy is adeno-associated virus, or AAV, and I'll talk about some of the challenges that are associated with these modalities. And finally, tools for demonstrating comparability, and I'll talk about some of the orthogonal approaches, along with some statistical considerations for demonstrating analytical comparability, followed by a case study of how to demonstrate that in case of empty capsids. But there are several other approaches that one may consider demonstrating such comparability approaches. Now, what is a comparability study? And I always like to ask three different questions: what, why, and how. Now, what is a comparability study? It's typically an impact assessment of changes that a manufacturing process might endure during the clinical development of a program. And for any modality, the changes are inevitable for process as well as formulation, as well as certain times there will be changes in the assays. So those changes are the reason where you want to demonstrate the comparability of pre and post change process. And many times these changes are defined as a major, minor, or moderate changes, or they're also called as a type 1, type 2 variations. And why do we demonstrate? Because typically in clinical studies, you want to demonstrate the pre and post change materials are comparable so you can continue the clinical trials. And how we do it, there are different strategies to do it. And also depends on the stage of the development of the product. If it is early stage, late stage, post approval, and also it depends on the different types of changes that are being implemented. So the design can vary depending on the application as well as the steps of the development. And typically, the comparability is a sequential process. The first step in the process is typically to show analytical comparability, which is mainly the comparison of quality attributes. And along with that, non-clinical comparability, where you may demonstrate the in vitro and in vivo comparability, along with PK/PD testing, and finally, clinical bridging. On the right, I'm showing a cartoon that depicts these three sequential processes. And one of the main aspects of comparability, like I mentioned, is analytical assay comparisons between pre and post change processes. That's one of the major studies you may want to conduct first, and before you may consider other options. And if you're not able to demonstrate the comparability, non-clinical study as well as the clinical bridgings might be necessary. I'm showing a couple of references at the bottom of the page, and those are very good references so for understanding the principles behind the comparability and also to approaches that you may want to use for in your own cases. Now, what is the analytical comparability process? So first and foremost, you want to determine what are the typical drivers for a change. Like I mentioned, mainly comparability studies are done if there are process or changes that are to the processes. And you want to demonstrate why, you want to understand what are the drivers for these changes. And these drivers typically either depend on quality of the product or commercial readiness of the product and process, as well as the presentation of the product. There are several examples in each of these categories that might be responsible or that might be drivers for these changes. For example, improving the process to reduce the impurity or improving the impurity profile or the purity profiles, and also improving the potency of the product. So these are some of the changes that you may want to be considering for process changes. Commercial readiness. And at some point, you may want to scale up the process or transfer to a commercial site. So those are also changes that may be necessary for the development of the product. And finally, the presentation of the product. So how the product is presented, is it a vial form or bag form, or also what is the volume of the product? So these are some of the drivers that you may also want to consider and to have a process that is commercial ready. And once you identify the drivers for your own processes, you want to identify and specify which specific process changes you will implement. And based on those, you may want to do risk assessment first to see what are the CQAs that are impacted by those process changes. And typically, these CQAs will be part of a safety, identity, safety, purity, and quality assessment of the profile. And based on these CQAs, you may want to write an analytical comparability protocol and assess how these CQAs will be impacted and what are the assays and what are the different tools you will use to demonstrate the comparability. And once you write the comparability protocol, it's advisable that you obtain the feedback from health authorities. And it's advisable to also get the feedback from multiple authorities so you can incorporate the feedback and revise the protocol accordingly. And once the protocol is approved, you may want to consider identifying pre and post change batches and continue to manufacture and release the product. And once you have a pre and post change batches available, then you will execute the comparability. And one of the primary sections of the comparability execution is the side-by-side testing. And once we execute those, you will want to consider the statistical evaluations and then demonstrate that those are actually equivalent. And finally, you will write a comparability report and submit the data in a regulatory submission to relevant agencies. Now, demonstrative comparability is one of the processes that actually requires teamwork. So that's a tagline here shown that it's a team effort that requires a teamwork. And there are several groups that are involved in this process. Even though analytical development group is the responsible and accountable group, there are several groups such as process development and formulation development who are responsible to understand the processes changes that they will be implementing, and what are the impacts of those on the CQAs that are part of the development process. And clinical manufacturing group will be supporting the manufacturing of the products from post and pre-change, as well as quality control, who will be responsible for testing these products and the GMP conditions. Regulatory groups will support the regulatory filing and overseeing the overall flow of the work, as well as the QA will again oversee the entirety of the process. A supply chain will typically help the understanding the availability of the material and managing the flow of the material towards analytical development group, so we can manage the study. So what are typical contents of a protocol? There are several sections in the comparability protocol, and one of the main sections is to demonstrate or to describe the history of the process and the intents of the change of the process, and also relevant of those changes to the process, including the risk assessment. And you want to demonstrate how the risk assessment decides on which CQAs will be impacted based on the changes that are being implemented. And the comparability strategy will include several different sections, and these would be looking at the release assays, stability, external characterization in process, and certain studies such as post-degradation studies you may want to include if you haven't had done previously. And so these are some of the sections you will want to consider having part of the comparability execution. And for each of these tests, you'll have to include justifications for each test, along with some of the criteria that you want to use to demonstrate the comparability. One of the critical parts of the comparability protocol is to decide on number of lots for pre and post change process. And due to the difficulties in manufacturing for gene therapy products, is one of the most important aspects of a protocol, that you want to ensure you have a sufficient number of lots to demonstrate the comparability. And you also want to share why you want to select those lots. So lot genealogy and also the history is also very critical to be a part of the comparability protocol. And like I mentioned earlier, that you have to demonstrate what methods you will be using. If there's historical data that you have, you may want to use to support the use of these methods, and also justification. And along with that, you will also want to include some of the acceptance criteria, and this can be quantitative or qualitative or semi-quantitative. Now, coming to gene therapy products such as AAV, there are several different challenges that we have to really consider in the comparability study. So far, these are still newer modalities, so there is limited understanding of these CQAs, and especially considering the different types of viruses such as lentivirus, AAV, and so there are differences to consider for these CQAs. And looking at the cell therapy versus gene therapy applications, there are several differences in the manufacturing processes. And along with that, there are compressed timelines for development. So that doesn't allow enough understanding of these products and processes. Due to the, again, shortened timelines, the number of lots or the batches that are produced will be generally low. So pre and post change number of lots will be always a difficult question to answer. So you have to really make sure that you have enough of these lots to be included in the comparability. And one of the main critical sources of issues could be the not having a representative material for some of the assays, for example, potency or dosing assays. So that can be a potential issue for these studies. And there are several CQAs for AAV products, and I'm showing you 4 different types of CQAs. Typically, we can divide them as physicochemical, biological activity, characterization, as well as purity or impurity. In the physicochemical, we look into physical properties, along with the physical titer, which is a typically genomic titer, which is a dosing assay, as well as the capsid titer. In the biological activity, we look at the infectivity of the virus as well as the expression of the candidate protein or RNA, and finally, mechanism of action reflective potency assay, which is also required to demonstrate the efficacy of the product. And in the purity and impurity, there are several types of CQAs, such as capsid DNA purity, along with the impurity that come from wholesale or process-related impurities. And you have to demonstrate all of those for these products. And one of the main sections of AAV drug development is to understand the product, and the characterization aspects of these products is very critical. And you will have to include several of these methods in the comparability. And some of the CQS that are shown are here that we need to demonstrate non-target DNA that is capsidated in the AAV molecule, along with the VP ratio and their modifications of the peptide sequences. So during the development of the product and also processes, it's inevitable that even the methods also change. Like I mentioned earlier, it's a combination of all processes and assays that they evolve during the manufacturing of the product and also clinical development of the product. So this is a typical flow of the assays that you would sometimes you'll have one assay that is typically used at early stage, but you will follow up with the development of multiple assays during the development, and you may want to choose the later stages of the assays to switch to switch the new assay or use them as a characterization assays. I'm showing you some examples on the right in the table that are typically have been seen in the gene therapy development. One of the primary ones that many of you might be already aware of it is switching from qPCR to ddPCR for titer, as well as empty full capsids that typically were measured by 8 to 60 to 80 or capsid to VG ratio. But there are many other methods that are now available that are shown here. So it's inevitable that methods also will change, and how do we incorporate these when you want to consider the comparability process. So as in a case study, I want to talk about empty capsid as CQAs. So as we all know, the manufacturing of the AAV product is complex, and typically there can be 4 different types of products that can be part of the manufactured product. And these could be either empty capsid that do not have any genome, while our target molecule is the capsid containing the full genome sequence, but that's not always the case. You may also have sometimes partial DNA package in the AAV, along with a non-target double-stranded or single-strand DNA that could be packaged in the AAV products. So these are complex situations in the manufactured product, and right now, we'll just focus on empty capsids as one of the modalities to show the comparability. And why these empty capsids are important? They can act as a decoys against the neutralizing antibodies, but also potential immunological consequences. And if the percentage of empty capsid is very high in the product, that increases the production cost of the material, and you'll want to have many more lots to supply the product to the patients. So it's very important to characterize these products and also to ensure the batch-to-batch consistency. And typically, ideal cases would like to have each capsid containing 1 genome, so the capsid to VG ratio should be 1. But that's ideal, but it's not the case generally. And there are several methods that are typically used even nowadays, are shown here. Some of the methods that are earlier that were used were based on optical density or calculation based on capsid to VG ratio. But there are many new methods that are being currently used. Most prominently, cryo-TEM for imaging empty and full capsids. AUC is the gold standard for looking at the empty, partial, and full capsids. Chromatography, especially anion exchange chromatography, has been routinely used now in the quality control as well as analytical development groups because of the fast turnaround times and QC friendliness. But along with that, there are methods such as mass photometry, which use the interferometric scattering mass spectrometric analysis to show empty and full capsid separation. And finally, CDMS, which also looks into native ion mass to charge ratio to really understand empty or full capsids, along with providing their entire masses. So each method has advantages as disadvantages, and not one method can give a truly understanding of the CQA. So it's advisable to look into multiple methods during characterization of the product as well as the comparability. So I want to talk to you about some of our own approaches that we have used. We have used the anion exchange HPLC for separating and quantifying empty and full capsids. And we published this paper a couple of years ago, and we've shown that we can demonstrate the separation of empty capsids from filled capsids with a very high resolution and very low tailing. And these are typical requirements of USP guidelines for a chromatographic method validation. So here I'm showing an example where empty capsid along with the full capsids or the mixture of these two have been successfully separated with high resolution. And using the peak integrations, we can easily calculate and measure these empty percent concentrations to very high accuracy and very low detection limits. Now, as an example, I'm showing you the data from 5 different AAV production lots from process development, along with a control AAV product, which should not have any package sequences. And on the left, I'm showing you the data from capsid to VG ratio. So if you look at that, based on how the VG titer is measured, either qPCR or ddPCR, it can give a very different perspective of how these lots can be compared to each other. And regardless of that, the ratio of either 0.5 or anywhere close to 2.5, it indicates it's impossible to have these ratios to be real indicative of the empty capsids in a product. So in a typical sense, the capsid to VG ratio is always going to be variable because it relies on 2 different assays. And so it may not be really reliable for a long-term solution. Now, there are several other methods that I showed you in the past slide, and we have shown the data on the right graph here. This includes AUC, TEM, CDMS, HPLC that we do in-house, along with the mass photometry. And looking at this figure, it may appear that many of these, lot if not all of these, would be most likely compatible, as they show a very similar profile of the empty capsids. So it's important to use all of the available assays or tools to really make a judgment on the comparability of some of these lots. Now, what are the different approaches for demonstrating comparability? And first and foremost is looking at the data that you have, looking at the distribution of the data, looking at the patterns, and also fitting a normal fit curves or best fit curve. So it all depends on the data as well as amount of data you may have for a particular CQA and assay. And one of the easiest ways to show the comparability is also to look at visually. And you saw previously in the graphs that in using VG to capsid ratio, it's very difficult to demonstrate they're comparable. But we're looking at the other assay, it may appear that those lots might be comparable. So this is one of the ways to really start looking into comparability, to visualize the data in itself. And you may want to identify obvious outliers based on visual inspection. And then you can also correlate the data from multiple assays for some attribute. And you'll see some of that in the next slide. The next approach is to use equivalence testing. And in this, typically, you would want to define a EAC, which is the equivalence acceptance criterion, which is nothing but an appropriate difference based on subject matter experts' opinion. That difference may practically be 0, so that then you can say that these lots are comparable. So it's important to define the equivalence acceptance criterion before you start a comparability, and that needs to be based on your previous data and from the process development or any other representative manufactured lots. And another option that I'm showing you here is to use the quality ranges. So you may want to consider this as an option for quantitative assays. So for these, you want to have some historical data. And once you have historical data, you can use some of the statistical multipliers, such as standard deviation, K sigma, C factor, or any other multiplier that is more representative of your data, and then use that to define a range. And the range can be one-sided or two-sided range depending on the type of the assay. Typically, for a purity or impurity test, it's fine to have a one-sided limit for the range. But for the critical quantitative assays such as dosing assays or potency assays, it's best to have a two-sided ranges. So how do we compare the orthogonal data from multiple assays? So here I'm showing comparison of our in-house HPLC method versus of all four different types of characterization methods, such as CDMS, TEM, AUC, of mass spectrometry. And if you look at all of these correlations, and these are bivariate fit analysis, if you look at all of these correlations, the correlation coefficient, it is shown at the top left of each graph, shows to be at least over 0.9, which shows the very good correlation. The best correlation we find is with the HPLC against the CDMS. So this shows that the data are very highly correlated with respect to all of these different methods. So that also justifies the use of HPLC method for in-house characterization. So you can correlate these methods later on in your comparability, or even if you're later on regulatory filings. And as I mentioned earlier, looking at the data is very critical. So you want to see how does the data look like, and what are the best fit curves, or what is the normal fit curve. And since these data are for empty capsids, and generally empty capsids are at the lower end of the measurements, so it's not uncommon to have not normal curve fits. So in here, I'm showing you each of these data sets, and the normal curve fits are done along with the best fit curves. And in all of them, you see that the best fit curve is exponential, which is not uncommon for measurement that are at the lower end of the curve. So it's important to do that for your data analysis and see if your data is normally distributed or not normally distributed. If the data is normally distributed, you may want to use additional tolerance intervals or similar statistics to really define the ranges. And looking into the comparability, like I mentioned earlier, that our goal is to show or to identify if these lots are comparable, or how these assays interact with how these assays are correlated with each other. So on the left, I'm showing you a graph of where we are plotting empty capsids of each of the lot, but from multiple assays as the data as aggregate data. And on the right, I'm showing a similar graph, but with respect to an assay, but from a different lots again. So here it can be seen that on the left, all the lots typically have a means which are very close to each other by using different methodologies. And the graph below shows the scatter plot for equivalence testing. I mentioned earlier the EAC criterion. You have to define that upfront. And I'm showing example criteria here, which significance level is around 0.25, so the confidence level will be around 0.75. And with that, if practical difference 10%, if that's practical difference is 0, that means looking at all of these assays, all of the lots, if there's a 10% difference is negligible, and then you may consider that as a comparable. Using that, the scatter plot shows that all these lots would be considered as equivalent. And on the right, looking at the different methodologies, it's very obvious that TEM gives a little higher imprecision, which is not uncommon given that it's an imaging technique, and there may be some errors because of the way the images are taken or quantified. However, all of the other methods show very high precision and also accuracy of the data. And more importantly, you can see that the means of the data from AEX, AUC, and CDMS correlate very, very highly. In respect to mass spectrometry, some of the resolution challenges might be there, but it also offers an additional layer of characterization that is important to have it. And using these data, again, equivalence testing, using the 10% as a practically negligible difference, you can see that all these technologies also show comparable data for all of these lots. And summary, finally, I want to leave you with some of the thoughts. And I also have a cartoon on the right to show the two different types of apples, two different colors of the apples, whether they're comparable or not. I'll leave that to you to decide. Because comparability is a demonstration of how the process changes, or how two products are comparable to each other or not. There are different CQAs, different aspects of comparability you have to predefine in your protocol. And like I mentioned earlier, the comparability is an integral process, so inevitable process for development as we go through manufacturing changes throughout the development process. The careful planning and execution is critical and also important to the success of the program. And I showed you that there are different types of CQAs, but also for each CQA, there may be multiple assays that you will want to consider to demonstrate the comparability. There are different ways to show the demonstrate the comparability. Use the individual assay data itself, or use the combined aggregate data approach that I showed you earlier. And it might be necessary to identify outlier analysis, and these are not uncommon. And for that, you have to have a sufficient number of data points so you can really identify and also eliminate the outlier analysis with proper justification. One of the most important advice I've gotten, I want to give it to you all, is seek help from experts in the field and to get it right. It's an important process, so you want to really get all the help you need. And with that, I want to acknowledge my team members at Sangamo Therapeutics, Analytical Development Group, and along with the process development, formulation development, and also several members from Vector Core and PPRC committee for reviewing the presentation. With that, I would like to hand it back to Marcus.
That's great. Thank you very much for that, Santosh. Great start to our webinar today and a great presentation. So thank you. And I'd just like to remind our audience members, if you have any questions for Santosh, I can see some coming in already. Please feel free to send in any more questions if you have any. And please also include it in your questions if they're for Santosh or if they're for James, okay? So we will be having our live Q&A session after our next presentation coming up. So we look forward to going through your questions then. Okay, so now please allow me to introduce our next presenter. So we have James Wilkinson, and James studied a BA in Natural Sciences, followed by a PhD in protein chemistry at the University of Cambridge, studying the biophysical properties of ankyrin repeat proteins. After a short postdoc, he joined Nanotemper Technologies in Munich in 2012, when the company was in its infancy. And as one of the first applications and sales specialists, he stayed for 6 years developing the technology, launching new products, and ended up running the UK subsidiary with responsibility for the UK and global distributor businesses. After working for a number of other startup companies, he joined Refeyn in 2019 as the sales director and now vice president, VP of sales and customer success. Refeyn are an Oxford spin-out company and have recently developed a methodology called mass photometry to analyze biomolecules. James is focused on introducing the technology globally and supporting our rapidly growing customer base. And with that, I'd now like to hand over to our next presenter. So welcome, James.
Thank you, Marcus. Thank you for the introduction. It was interesting hearing that, because I think probably needs rewriting. So as I said, I joined Refeyn when we were a very small company. We were about 18 people, spin out from the University of Oxford. We have grown rapidly over the last few years, and I think that the last count, we were about 160, 170 people globally. So we have a lot of things going on at the moment, and one of the things I'd like to talk to you about is one of our relatively recent developments, which is using mass photometry for the characterizations of AAVs. So before I start, I'd like to just acknowledge a couple of people. So my colleague Svea put these slides together. So she's responsible for any little spelling mistakes. Unfortunately, she can't be here today. She's a little bit ill. And also thank you in advance to Santosh. There's quite a number of topics that you introduced very eloquently that I will be taking advantage over the next 30 minutes or so. So what are we trying to do? We're a company that developed a technology called mass photometry. Mass photometry was launched and was sold very successfully as a very new method for protein analytics. But we're always trying to innovate. We're always trying to look to see where this technology can be used further than its initial starting point, which was protein analytics. And we came across this problem that Santosh has been talking about. So specifically with the AAVs, but this is valid for a lot of other vectors and other particles as well, is there's a technology gap here. So we like to think about things from a technology gap and work upwards then to see how we can solve that. So if we look at the particles on the left, very clearly we have an empty particle and a full particle, and these have a very different mass. And mass photometry measures mass. So we thought we could start getting involved with this. And we also started to think about what the perfect analytical solution would look like. So our instruments, and this is a core across our whole instrument range, we work with very fast measurements. They're very easy to use, and they use extremely low volume samples. So these are all things that you would have as a tick box for your ideal AAV analysis method. And we also have our instrument and our technology dressed up in benchtop instruments. So you do not have to send these off to partners to be measured. It means the turnaround time, which we've heard a lot about, is extremely rapid, minutes it can be. And the reason we're trying to solve this problem, I think we've heard a little bit about before. If you don't know what you've got, you're at risk of increased immune response, you're at risk of increased costs, for example, capsid precipitation, aggregation, all of these bad words that we want to avoid. So just to introduce our technology, basically when we first launched this in our first instrument called the 1MP back in 2019, technology works fundamentally in the same way. We have our particles in solution. Now this can be AAV capsids, this could be a 30 kilodalton protein. These proteins will then bind to a glass cover slip, and we shine some light at them, and some of this light is reflected off the particles, some of this light is reflected off the glass cover slip. And both of those portions of light we interfere, and we get these interference patterns, and these cause these spots in the middle on the left-hand side, these concentric rings. And the key to our technology is the contrast that you're seeing, i.e. how dark the dark is and how light the light is. That contrast is directly proportional to the mass, the molecular weight. So what we see here on the bottom left is two populations, two different masses, which our software then automatically fits with Gaussian distributions, and we can start seeing two different species. Now, this was something that was able to be measured. If we took AAVs to our older instruments, the 1MP and the 2MP, we could see AAVs. But because this problem was so big, we decided to optimize our mass photometer for this system specifically. What this means is the SamuX system, which is what we call it, the SamuX will not measure a 30 kilodalton protein, which is really optimized for the AAV mass range. So it's optimized for the megadalton range, and that gave us a much higher resolution than we had on our old mass photometers. And you can see and hopefully agree that we're seeing these two very distinct peaks, baseline resolved, one at the mass of empty AAVs and one at the mass of full AAVs. I'll go a little bit more into that throughout the talk. So this is the instrument. For those of you who might be familiar with our other instruments, it looks very much like those. But as I said, optimized for that 3 to 6 megadalton AAV range. The other changes we made are to make it more suitable for the AAV workflows. So we optimized the hardware for decontamination procedures. And then also we have dedicated software, which runs on the SamuX for measurement of empty-full ratios, for example. So this allows you an extremely quick turnaround time. It allows you measurements in a couple of minutes using very, very little sample. And it is really very, very simple system. Something I'll touch on at the end, but just to introduce now, this system is now available as well. This is our auto Samux. So it's the core of it is the same Samux that you will have seen and I'll talk about, but it's inside a robotic system which allows a walk-away time of about 1.5 hours and the autonomous measurement of 24 samples within that time frame. So this is really important for automation. If you've got multiple samples you want to measure, it's also very easy to use, but the key thing also is it's really reproducible. No matter how much we like to think of ourselves as experienced pipetters, the robot will pipette better. So it's a little bit more accurate in the pipetting of this as well. So this system is available for order now. It is also an upgradable option on the existing Samux systems. We're still a relatively young company and it's important to us to keep our customers very happy. All of the existing Samuxes that are out there since that was launched a couple of years ago, it's important that we offer the upgrade to those customers as well so that they can take advantage of this new flexible system. So I'd like to spend most of the talk sharing with you some data. This is all now measured on our Samux, so the system which is optimized for AAV analysis. So what you see on the left-hand side is the output from 4 or 5 different measurements. We're showing where we've mixed commercially bought mixtures of empty and full vectors. So we simply mixed different proportions of empty and full, and then we made the measurements on the Samux system, and we can see on the left decreasing peak size for the empties around the 3.5 to 4 megadalton range, and an increasing size of peak for the 4.5 to 5. Now the important thing to note about mass photometry is we are a single particle counting method. So these are not just arbitrary peaks. These are not peaks that are fitted through some absorption data or something, for example. These are made up of individually counted particles. So we would typically measure a couple of thousand particles in a measurement, and they then go towards forming that Gaussian. So it's an extremely accurate method, sensitive method for picking these things up. And if you look at a plot now of the loading measured by mass photometry on the right-hand side, against on the x-axis the predicted loading based on our mixing, you can see an extremely linear response as you'd expect. The other thing to note though is there are triplicate error bars within these, but they're smaller than these data points. This really shows this high precision of measuring single particle. We were new. Our main conclusion is that we can consistently perform across the entire range from very, very low to empty all the way up to almost full. Now this system has been on the market for a while, but prior to that and also during that early phase, we had a lot of partners which were very kind to test the system for us and give us a lot of feedback. So this is all data courtesy of Victor and Professor Albert Heck in Utrecht. One of the questions we had very initially is, great, we can measure the AAV8s on this, but can we measure other serotypes? Now we know, of course, there's lots of different serotypes in use for different reasons around the world, so we wanted to make sure our technology was compatible with all of these. What you see on the left-hand side is mass photometry measurements that we measured in-house, using just 4 different serotypes there. And again, it was very, very nice peaks. These are all empty, just for reference. These measurements or similar measurements were then repeated in Utrecht using different serotypes and also different serotypes from different companies. So they've gone through different production runs, different purification methods, and we're all getting very, very nice clean peaks. The really exciting thing about this slide, though, Albert is a very experienced mass spectrometrist, so they do a lot of CDMS, which you've already heard of. So they could compare our measurements with their CDMS data, and they were all very, very comparable. The key thing also to note is we are really measuring the mass here. So not only is 3.81 matching 3.83, but actually 3.81 is giving you something. It is giving you the mass very accurately of this particle. So this is an extremely important point. Now, one of the big differences then between the dedicated Samux system and mass photometers of old, we can see these beautifully separated empty and full peaks, but we can also see in this data on the left-hand side some data in between these 2 peaks. So we were really excited about this because this is what we're seeing here is these partially filled particles. These are particles that have partial genome in, a degraded genome, other genome that they shouldn't have in, all of the above essentially. So not only can we see very clearly a nice peak separated for the empty, a nice peak separated for the full, we can quantify that again with a single particle counting. That quantification fits very nicely with the set miles data, which these guys had already done, but we can see these intermediate particles in between. So they then took these samples, extracted the DNA, and ran the DNA on a simple agarose gel, and you can see this in the middle here. So the full length cargo DNA is obviously the predominant band, which fits with our mass photometry data, but we can also see multiple shorter bands on the gel here. And of course, because we've got multiple shorter bands, we've got a range heterogeneous sample, which is what we see these partially filled intermediates on this thing. So this is all courtesy of a biopharma company we were with. Not only that, this I got very excited about. This is something that was actually very new to me. It took me a while to work out what was going on here. We're seeing now very nicely defined peaks to empty and full, as we should be used to by now, but we're actually in this set of data, both on the left-hand side and in the middle, seeing a peak at much higher masses as well. So what we're seeing is these overfilled particles. And we saw this data twice with two industrial partner collaborations we had. We focus on the one on the right-hand side. We see these very nice three needles of empty, full, and overfill, and they're evenly spaced. So if we measure the mass of them, they were approximately 30% each, a little bit more on the full side, but the difference between the empty and the full, mass-wise, was identical to the difference between the full and overfilled mass-wise. So we're measuring the mass change, where this AAV will go from an empty to a full, and a full to what we're calling an overfill. And talking with the collaborator, this is exactly what was going on. They ran other gels and other methods as well, and they'd see that in the overfilled capsids, it actually had 2 copies of the gene of interest, and this was by design. The full one was only registering 1 copy, so it really was twice the amount of DNA they were packing. But again, hopefully you can see how beautifully separated these peaks are, and by measuring the exact mass, you can also start to quantify exactly what you've got. You can check whether you have the DNA that you expect to have produced in the capsid. Now, a final feather in the hat, if you like, for the SAMUX and mass photometry is being able to give insights into aggregation. So I didn't mention the specifications of the system earlier. The SAMUX is optimized for the megadalton range for AAVs, and it will measure between about 500 kilodaltons and 6 megadaltons. That's the range where we can very accurately determine the mass. Seeing larger particles, though, is fundamentally easier than seeing smaller particles. So we do see larger particles on the SAMUX, up to 20-25 megadaltons. Quantification of these populations, though, we haven't yet tested and validated. So we can see them. We're happy they're there. We're happy they're approximately the mass we're saying, but just take them with a bit of a pinch of salt above our sort of 6-8 megadalton rate. However, what we're seeing in this data is different populations at suspiciously different areas. So if we take a single AAV, we're seeing that as a nice defined mass here. What we also see is a large population, and these are individually counted particles. Remember, we've seen a single particle, we've measured its mass, and we've put that into a histogram with others of the same sort of mass. And this represents an aggregate. This, in this situation, represents 2 AAVs that have come together. Population up here represents 3 AAVs that have come together, etc., etc. So it's not as quantitative as the MT4 measurements, for example, but we're really getting insights into aggregation there. You can see, therefore, different drying methods, different reconstitution methods, being able to give you different proportions of aggregation, and all the way up to approximately 5 AAVs. We can see. So this was something that was courtesy of Catapult Cell and Gene Therapy. So this is a UK-based, government-funded sort of innovation hub, and a pharmaceutical company called Pharmaron, based in Liverpool. When we started digging into this, we noticed that there are 2 real gold standard methods, if you like. These are the methods that seem to be the gold standard used by lots of people all around the world, and for good reason. So one is electron microscopy, specifically cryo-TEM, and the other one was analytical ultracentrifugation. Now, I've used both of these methods myself for AAV measurements, but also lots of other things. They're great methods. What they are not, though, is quick and easy to use. Cryo-TEM requires an awful lot of infrastructure. The equipment is obviously very expensive. The sample preparation takes a little while. AUC takes a while to run, and the data analysis still gives me a few nightmares from the days as a PhD student. And this is where we designed the Samux. This is the technology gap that we saw. The Samux is exceptionally easy to use, a couple of minutes. And the data that we got out from both the Catapult and FarmOne really gave us a lot of confidence that we were measuring the same thing. They were really in good agreement. So I won't go through all the numbers. This data is on our website. There's an application note on this, so please look forward to it. But yes, we did the benchmarking against these two methods, and we were comparable with those, what we consider the gold standards. So they also, and this is also done in FarmOne, just as a reference, this is not representative for FarmOne's platform process. This is something that they did in collaboration with us, just to show how the Samux can really be used in this process, is using it to optimize your purification protocols. And the reason you can do this is because we can measure quickly using very little sample, and also on the bench. So the turnaround time is extremely quick, and the measurement time is extremely quick and cheap. So what we have here is the clarified AAVs right at the beginning, and we've got all of these different fractions then throughout the purification process. We've got the flow through, then the first elution, second elution, and on. And what we can see very quickly by making all these measurements is that actually elution 4 is most enriched for full AAV capsids, which is this blue section. So this allows you to then start thinking about playing with this process. So here we've got 7 different elution steps. Now, this led us to the development of the Samux Auto. So as soon as you're starting to make more than a couple of measurements, it really benefits from being able to have an autonomous measurement of 24 samples. It's operator free in that time, so the operator can go away and do something else. You can come back an hour and a half later to all of your measurements being made. The data is all analyzed in the system as well, and it's extremely easy to use still system. If you look at this middle picture, you put your samples on this holder here. The Samux is actually on the left-hand picture in this bottom tray hidden by the blue. So the sample shelf is this part in the middle. You can just see it in the middle picture on the left-hand side. And the robot will prepare from your sample plate onto the system, make a measurement, and then continue up to 24 samples. So I'd like to finish with a couple of case studies where now with this extra capability of the Samux Auto, with the automated measurement of 24 samples, we can really make the best of this system and improve data, get the workflows all working really nice. So what we have here is a multi-step elution test. So these guys were eluting their systems with, forget how many different steps, but you can see the multi-steps here, many steps of elution. What they were then doing is on their chromatography system, measuring by UV, so looking at 260, 280 for the DNA protein ratios. So what we're looking for are numbers above 1, so above 1.1. So above 1.1, it's mainly full, we've got a lot of DNA in there. Below 1.1 is mainly empty, not much DNA in there. So this ratio on the right-hand column you can see here, and we've put a box around the samples from 5A2 to 5A5. These are ones we've then gone on to measure with mass photometry. You can see the greatest percentage of falls predicted is this 1.127. So this is a multi-step process, all of these different fractions, and we're now measuring these fractions from a ratio of 0.46, which should be quite empty, up to 1.16, which should be quite full. And this is, no surprises, what we see. The A2 one, we see really mostly empty. What we do in our software is we set gates, and we can then count all of the particles in this light blue, all of the particles in the darker blue, and then create a percentage full ratio from that. So you see A2, all of it's in the left, very little, if not 1 or 2 particles in the right-hand side. A5, we've got a much bigger peak here, very few empty particles. So we're measuring ratios from 6% up to 88%. The A4 was the highest one. So this allows us to measure these again within 30 minutes. We're also getting additional insights here, and this is something that you were missing from your set UV analysis. We're seeing little bits of aggregation up here. We're getting insights into partially filled particles here. We're getting proteins or smaller species, giving additional insights into purity. So because we count every particle, we can see all of this, and we can see exactly what's in your sample, even if it's in an extremely small amount. So the next step was then to think about the two-step elution protocol. We had a multi-step strategy just before, 7 or 8 steps. Can we then use that mass photometry data to bring that elution step down to 2 steps? This makes the whole process a lot more efficient. So what we're now doing, we've designed this system where we're going up to about 20 mil, and then we start eluting what should be predominantly the empties, and then up to about 33, 34 mil, and we're eluting what should be predominantly the full, before we then elute final elution from all of the bigger. So the UV readings, if you look at the ratios, again, 0.65 and 1.17, so the UV readings suggest it's worked in that we've got mostly empties and mostly full. But let's now look at the mass photometry data. So the mass photometry data does indeed confirm that. We do have a significant proportion of full in this empty one, 20% or so, and the full is mostly, again, 20% or so empty. So they're almost mirror images of each other. So that's what you would expect. Now we've reduced that elution profile down from 7 to 2. However, interestingly, what we're seeing is the fast elution protocol has actually led to increased amounts of partially filled, if you look here on the empty, and also overfilled, much more in this sample of this overfilled bracket than we saw in the previous set of data. So that fast elution profile is actually causing an increase in partially filled intermediates to be within those elutions, and also an increase in the overfilled AAVs. Now, though neither of these things are wanted by this industrial partner, so this is essentially a contaminant within their prep. And if you imagine those other elution profiles where we had 7 or 8 steps, that's where those ones will be coming out. So by rushing it, in effect, with a 2-step elution, we're actually seeing extra things coming on. But this, importantly, is things that you did not see in your UV ratio data. So being able to see that single particle mass spectrometry data gives you those extra results. Now this is showing a similar story, but slightly different data. What we've got here is same vector. So this is an AAV8 vector, again produced by an industrial partner, and they're producing these in 2 different protocols, Prep A and Prep B. What they saw actually after the mass photometry data was when they looked at these things, the vector A was showing a much higher transgene activity compared with vector B. And this was identical titer doses. So the vector B was showing a much reduced activity. When you look into the mass photometry data and show what you've actually produced at the end of this manufacturing process, we look on the left-hand side, Prep A, getting very nice, clear, empty and full. When we look at Prep B, however, the distance between these empty and full peaks is just the same. The reason that it looks not quite as baseline resolved as A is because of all of these partially filled bits here. So what we're seeing actually is a lot of fragmented genome. Seeing an awful lot of the genome fragmenting, being packaged into particles and giving us this heterogeneous spread of particles somewhere in between what should be the true empty and the true full. So this was a nice set of data, again confirming that being able to do this analysis at all stages throughout your production gives you a lot of insights into what's going on. Now, the very final thing I'd like to share with you, this is something that's a little bit new, is we've introduced a titer estimation to both the SamuxMP and the Samux Auto. This is an estimation tool. We're very clear on that. This is in correlation to our very accurate mass. We're measuring very accurate empty-full ratios. We've done the comparison against simple ELISAs. The comparison is very good. there is now a button, or going to be a button on the software, which will allow you to make this titer estimation. But it's not, we're not claiming it's as accurate as our empty fill ratios, for example, but it's going to give you very, very good data, all within factor of 2 relative to the ELISAs. So this should again bring another branch to the instrument, being allowed you to see the titer as well as the empty fill ratios, as well as any partials, as well as any aggregation, all from that one very quick measurement. And just a little bit of a preliminary bit. We are, as I'm sure most of you are aware, working on our GMP regulated compliant software. This is something which we aim to launch in Q3 this year, so actually not too far away. This will be upgradable, of course, on all the existing systems. And this will then allow the SAMUX system to be really used throughout the process. So from the R&D all the way into PD labs. So this, reach out to your reps if you're interested. But if you're new to the systems, we now have the SAMUX in a fully automated walk-away autonomous system with GMP compliant software as of Q3 ready. So just to summarize, hopefully I've convinced you. Data on the screen again now. We can see very clearly empty and full ratios. What we can also start to see is the partially filled intermediates in between. The SAMUX Auto now allows new measurements, new assays, new workflows through the autonomous measurement of 24 samples. And through our GMP software, which is coming, should be applicable at various different purification stages, all the way through from R&D to manufacturing. This, again, the measurements are very, very quick, take a few minutes. We seem to be serotype independent, and cost-wise, really, really minimal cost, both in material as well as consumables from our system. So, my acknowledgements are simply all of Refeyn. We have a rapidly growing company with amazing colleagues, and we've grown very quickly, and an awful lot of people have contributed to this, as well as, of course, our industrial partners who developed this system. So thank you very much, and I'll be very happy to take some questions.
That's great. Thank you very much for that, James. I just second James's takeaway comment there. If you have any questions for him, then please feel free to send them in via the questions tab located directly below your webinar screen. We look forward to answering your questions in our Q&A session, which is coming up in just a moment. So thank you once again, James, and thank you as well, Santosh, for your opening presentation. And I'm just going to bring us back to the camera feed now, okay, before we start our Q&A. So one moment. Very good. Okay, great. Thanks a lot. So our first question today will go to Santosh. And Santosh here, your question says, what are some examples of changes that would require a comparability study?
There are several examples I can speak of. One of the primary examples is to improve the impurity profiles. So if you are concerned about a specific impurity in the process and you want to remove it, then you will consider implementing process changes. Other example would be increasing the concentration of the product, or changing the manufacturing site from one region to another region, a different country. So I think these are the typical examples you will encounter as you go through the development process and early to late phase, or even near commercial. We want to think about these changes that will be inevitable for the process.
Thanks a lot for that, Santosh. Before we move on to our next question, just had to quickly check. James, was there any additional comments you'd like to give to this answer?
No, not at all. It's more complete than I could have given.
Very good. Okay, thanks a lot then. Our next question for James, it says, how is the system calibrated?
Yeah, good question. So we're actually just about to launch a calibration for it, but essentially we measure contrast. That's what we do. Contrast is proportional to mass, so we need a way of working out how proportional. So at the moment you can calibrate with anything, any proteins, any AAVs. The calibration we're launching is a high mass, specifically the SAMUX, using AAVs. So basically you measure the contrast of a known mass, and that gives you a linear relationship to determine the mass of a known contrast. So it takes a minute. So this is a simple measurement.
That's great. Thanks a lot for that, James. And Santosh, any additional comments there?
No, I think that's good.
Okay, thanks a lot then. Our next question for Santosh says, how long before you plan to start the comparability study?
I think the easiest answer would be as early as possible, because these are long drawn processes, especially writing a protocol, having agreement within the technical operations division or in your company itself, and then following up with the regulatory agencies, getting their feedback, and revising the protocols. So all of these can be long drawn processes. So it's advisable you start as early as possible, and have a plan ready, as you intend to go to your next development of the process. So that's the short answer I can give to that.
Mm-hmm. Thanks a lot for that, Santosh. And James, any additional comments there before we move on?
No.
No? Very good. Okay, thanks a lot. Next question for James says, is there a plan to support high throughput screening in the future?
There's always plans. So our first stage was to get the system in use and in regular use, and we've succeeded in that. The demand, therefore, has been for more measurements than one. Again, I think the system is quick enough, where we have had the case that it's too quick to get your lab coat off. So we now have the Samux Auto that will make measurements of 24 samples away. After that, yes. So there will be high throughput systems to come for AAV analysis and also mass spectrometry generally, but not today.
Thanks a lot for that, James. Santosh, any additional comments there?
I think I would love to. I'm looking forward to those high throughput systems. So from Refeyn. So
yes, no, I think that the demand is there. I think, it needs to be high throughput. We deliberately don't call the system a high throughput system. It's an autonomous system. It allows you to do some repeats, etc. But we appreciate that 24 samples is not close to the high throughput. But yes, we will be working on a solution, and that will be coming over the next few years.
Very nice. Thanks. Our next question for Santosh says, do we always need to show comparability in analytical, preclinical, and clinical, or we go to preclinical and clinical when analytical comparability can't be proved?
I think the first process is always to generally show analytical comparability, where you would compare the QCAs and perform several analytical tests to show that pre and post change process materials are comparable. And if for some reason you are not able to show that comparability, you will take advisement from health authority to see what are the additional approaches. And those non-clinical and clinical approaches are the subsequent approaches that you may consider. Again, I want to remind that those are long drawn, cost and time prohibitive, and also cost intensive processes. So it's best to put your best to have your plans ready for analytical compatibility and show that you can demonstrate that before you're thinking about other approaches.
Thanks a lot for that, Santosh. And James, any additional comments there?
No, other than from an instrument manufacturer's perspective, I think this is what's so exciting. There's so many new methods that can do lots and lots of different things, and comparability is the key word. You need to be confident in your data. For that, you need multiple methods.
Very well said. Thanks a lot. Our next question then for James says, how does the titer measurement work?
Oh, the titer measurement. So it's a combination of we have our calibration. So we have to run a calibration as part of any titer measurement. We know how a calibration system behaves. We've also programmed a lot of the software and the learning for this, what we expect to see in terms of particle count wise for different concentrations. So we've done various titrations ourselves, and that then outputs an estimated titer from the custom sample. So it's fairly straightforward, which also means it's fairly estimation rather than real accuracy. But frankly, a lot of the other methods to do it, they might work. If you think about an ELISA, plus or minus a significant amount, we're in that ballpark. So I think it's a good addition to the system.
Thanks so far, James. And Santosh, any additional comments there?
No comment on that.
Okay, thanks a lot. Our next question for Santosh says, what is a sufficient number of lots for comparability?
It's a very good question, and I think typically, it's considered good to have at least 3 pre and 3 post change lots for comparability study. Again, you have to justify which lots you are selecting and how many lots for each of these pre and post change, and get the feedback from regulatory agencies. But this is generally what is acceptable, and what I've seen from discussions with others, that this will be the minimum requirement. But again, more the better, because that also adds up to your statistical considerations. And depending on what type of statistic you are using, you may want to include additional lots, whether they are clinical lots or they are process development lots. Those always will be helpful to include those. So you have more statistical considerations in your comparability.
Thanks a lot for that, Santosh. And any additional comments there to add, James?
No, I mean, so yeah, more data is always good. And I think that's one of the key things we're trying to do, the ability to allow people to create more data in a short period of time.
That's great. Thanks a lot then. Our next question for James says, can in-process samples be measured, or does the sample need to be purified?
Good question. They can be. I've seen them measured. The limitation, if you like, from our technique is that we see everything. So we're a single particle method. We will see every particle in that sample. The good thing about this specific application is these particles are very large. If you were trying to see 200 kilodalton molecule in the mess of everything, it would be difficult. Seeing a 3 megadalton particle is very visible. So yeah, it's not an easy question to answer. Generally, we've measured it. You will see the background of everything you've got. If your AAVs are there at the size range, you will see that. But it's not something we've worked too much on and validated on too much.
Thanks a lot, James. And Santosh, any additional comments there?
I would echo James, saying that sample quality is a critical component of the any analysis. So having minimal interference for evaluation by any technology is always helpful.
Yes, no, agree. And I think that's what you get. Problem is, every purification step you do, all of these things takes something away. So if you can measure in the more native inverted commas environment, the better. And yes, I think we're onto that, but it's going to take a little bit of development.
Thanks a lot for your answers there. Our next question for Santosh says, can you use lots designed for comparability protocol for clinical use as well?
Yeah, I mean, because when you define the number of lots and also types of lots that you will be including in comparability, you'll have to show why these lots are suitable. And if those clinical lots have been used in the clinical studies, more or less likely you'll have to include those in the comparability. And you can also include the development lots if you have additional lots that are representative. But I see that you could have a combination of both the clinical and non-clinical lots to be a part of compatibility.
Thanks a lot, Santosh. And James, any additional comments there? No, very good. Okay, thanks a lot. I think this is a question for James, but feel free to defer if you want. So it says, the low resolution for partial capsids is a technique limitation, or do you think the methods can be improved to be able to separate partial capsids?
So I'm in the fortunate position of not being an R&D engineer. So my answer is always yes, it can be improved. Essentially, I look from where we've come from. Like the 1MP system, which is our initial mass photometer, could see particles at this size, but you would struggle to really see 2 peaks at empty and full. The 2MP, you could be very confident you were seeing 2 peaks, but they weren't that well resolved. The Samux, now we've got very, very well resolved empty and full peaks, and we're seeing it within the middle. If we can stretch that even further, and if you think back to that DNA ladder I showed you, we could see individual peaks for those. It's theoretically possible. How difficult is it? It's a different question, but we will work on it. If it's possible, we will get there.
That's great. Thanks a lot for that, James. And Santosh, any additional comments there?
I think that's one of the holy grails of question, to identify and measure partials. I think for several years, people were wondering about how to measure empty capsids. But I think for last several years, we've done some great, we had great technology that at least gave us some answers of empty capsid. But partial capsids, and there are technologies available, and like James said, those are limited by resolution, also the sample quality. If the sample does not have any partials or have low concentrations, no technology can detect that if they are not above the detection limits of the technology. So I think it depends on the sample quality, manufacturing process, and also the technology in itself. And with more advances that we are hearing, I think as I can see in the future, there will be some technologies, a combination of technology that will give us the idea of accuracy of these partial capsids.
That's great. Thanks a lot for your answers there. Our next question for Santosh says, what tool is being used to show the TOST results? Does it require coding knowledge?
The results I showed are based on a jump analysis. It's a software available commercially. So, but other than that, there are several other softwares available, and I don't think one needs to have a coding knowledge to really do this analysis, especially the quality ranges that one can do easily on using an Excel with the with a plus minus multiplier. But for equivalence testing, you will need some sort of software such as jump analysis.
Mm-hmm. That's great. Thanks a lot for that, Santosh. And James, any additional comments there?
No.
Okay, thanks a lot. And another question for Santosh here says, how do you decide if more than one assay are needed to show comparability?
It's a great question, interesting question too. So, if again depends on the how much knowledge you have for the process and the CQAs and the assays. But typically for newer modalities such as gene therapy or cell therapy products, it's very unlikely that a single CQA would be representing or single assay would represent the understanding of a CQA. So it's very likely that you will have to include multiple assays because each assay works on different principles, and those can give you different information. So it's advisable that you use many of those assays to really understand the different aspects of the same CQA. It's also good to have different options available just in case one technology or one assay is not able to differentiate the lots that you are trying to evaluate, if they're comparable or not. And if there are technologies that can show you the differences, and those differences can be minor, smaller, or they may not be really affecting the efficacy of the product. But as long as you have those technologies, different assays available that you can evaluate, several aspects of a CQA, those are much advisable.
Mm-hmm. Thanks a lot for that, Santosh. And James, any additional comments there?
Yeah, it's a very interesting question. I think what everyone has to remember, this is a relatively young field. I think relative to antibody development, small molecule drug discovery, things like that. So these are questions that are coming up. How comparable do they need to be for efficacy? How comparable are they being made through the technology limitations in analyzing them or in the production of making them? So it's an exciting time to be in this field.
That's great. Thanks a lot for your comments there. So I'll move on. Our next question for Santosh here says, TEM is usually considered as gold standard, but you mentioned that is a technique that has less accuracy based on slide 18. Can you comment?
I think that's a very good question. And so technology in itself, TEM or any other technology, they're great on their own, but they are also limited by the sample quality and what are the concentrations of the CQA that you are measuring. Especially the lower range of empty capsids, it's very hard to distinguish them from partials or even full capsids. So inevitably, every technology may have some limitations at the lower end of the spectrum. And what I showed in the slide is that we see some variability at those using the TEM. And TEM itself uses imaging and also understanding the density difference between electrons and whether they're present or not present to show this empty or full capsids. And there are some undefined capsids also. So that resolution can get pretty low, pretty difficult at sometimes at the lower empty capsid concentration. So that's a limitation, I would say, both on technology as well as the sample quality.
That's great. Thanks a lot for that, Santosh. And any additional comments there, James?
Yeah, so I think sample preparation seemed to be a lot, from my understanding, from TEM. I think the methods can be very accurate. I think if you send the sample to the same person 5 times, they should get very, very similar answers. Sample preparation in our hands seemed to be a very big thing. Very small differences in sample prep can make surprisingly big differences in your final number. So it's that human element in any of these measurements. You put the human element into it, and you get variability.
I want to add one more thing. So so that also buffer excipients that are part of the formulation. They can also interfere with the imaging technique. So that's also one of the considerations you have to give what type of formulation you have for these products and before you use for these technologies. If there are interfering compounds or molecules in your buffer, then you want to eliminate those before you send those for analysis. So I think that's one of the reasons also TEM can be challenging at lower concentration because most of these AAVs come the surfactant as a formulation component, and that can interfere at lower levels.
That's great. Thanks a lot for your answers there. Our next question to Santosh says, do you have to use statistical analysis to demonstrate comparability when you have limited lot numbers?
I think it is you'll have to show the comparability by using a certain type of statistical evaluation. If number of lots are low, you have to use appropriate statistical analysis. It becomes very difficult if you have a low number of data points. So you have to take into consideration the regulatory agencies' input and what are they recommending. And there are publications also that they talk about how to demonstrate comparability if you have a limited number of data points. And I can say that it will be very challenging. So it's always advisable to include a minimum number of lots that gives you some statistical relevance to the data.
Thanks a lot for that, Santosh. And any additional comments there, James? Okay, thanks a lot. Our next question for Santosh here says, do you have to show each assay result is comparable if you use multiple assays for single COA?
I think that's a great question, and it's unlikely that all the assays for same CQA will show comparability. I think because all the assays, like I mentioned, they use a different principle, and it's great if they show all of them are comparable, but it's very likely that you will see some differences. Again, the comparability demonstration depends on the how wide you set the range based on your data. If you set the range is very wide, then everything will be comparable regardless of what assay or CQ you use. But if you really define your acceptance criteria very appropriately, then you will see some differences based on these different assays. But as long as you demonstrate that those differences are not impacting the safety and efficacy of the product, and those you can explain in your regulatory submissions, that your justification. And more data you gather, these will help you in the future potential commercial filing also, that you understand the product better early on throughout your comparability, not only but also characterization of the product. So it's challenging to see to expect or to see all the assets for single CQA will be comparable, and I see most cases you will have some differences in one of the assays for each CQA.
Mm-hmm. That's great. Thanks a lot for that, Santosh. And before we move on, James, is there any additional comments there that you had?
No, nothing I could add to that.
Okay. Thanks a lot. Well, that was our last question for today. So thank you to all of the audience members who sent in the interesting questions. We had a nice discussion going on this Q&A sessions, and thank you. And if you have any more questions later, or if you're watching this webinar on demand and you want some questions answered, feel free to send your questions over to me and I will forward them over to our presenters who are here today. So my email address for you, I'll spell it, it's marcus.osei@biopharmawebinars.com. Okay, and just to remind you as well, you can watch this webinar and other webinars by visiting our website, and this can be found at biopharmawebinars.com/webinars. Okay, but before I finish today's presentation, I'd just like to ask our presenters if they have any closing remarks. So Santosh, over to you.
Thank you everyone for joining today. I hope I've given you some of the consideration for comparability that you will encounter at some point in your programs. And I will leave you guys with the last comment that I had in the slide, that there are plenty of resources and help available, and please do take them, and it will be well worth your time. These are complex processes, and any help that is given, I think that's will be really beneficial for your success.
Thanks, Santosh, and over to you, James.
And yes, it's a pleasure to be invited. So from the instrument manufacturer's perspective, it's always great to see these systems being used. but B, also great to hear about challenges. You know, the way we see our job is we want to solve these problems. So please do tell us your problems. We've got a whole bunch of R&D engineers ready to try and solve these things, and if we can try and make these things even higher resolution for you, we will.
Very well said. Thank you. And I'd also like to take this opportunity to thank our presenters here today. So once again, thank you. That was Santoshkumar Khatwani, Senior Director of Analytical Development at Sangamo Therapeutics. Thank you very much for joining us today. And also James Wilkinson, Vice President of Corporate Development at Refeyn. Thank you as well, James, for joining us. It was a pleasure to have you both here. And it was a very nice presentation today. So thank you very much. Looking forward to what comes next. Okay, we're all done here now. I'm going to end the webinar. So have a great day, everybody. Okay, thank you.
Thank you.
Bye.